Thanks for everyone for coming to hear us talk about our work that we've been on for the last couple of years. So yeah, I'm going to talk to you guys a little bit about radio tracking monarchs and our work doing that to harness that technology to unravel the Western migration and provide a little bit more insights on their habitat that they use. So I just want to give a little bit of a roadmap on the different points we're going to be hitting in this talk. So first, I'm going to give you guys a quick crash course on the background of Western monarch biology. Next, I'm going to give you guys a quick intro to radiotelemetry and animal tagging in general. And then we're going to review what the MODIS network is. And then we'll talk about the work that we've been doing here in future directions, along with a lot of our partners. So, jumping right into the background of monarch biology. So, the population of North American monarch butterflies, as you guys might know, is roughly bifurcated into east and west by the Rocky Mountains. So, in spring and summer, which is mostly the green and yellow areas on this map, monarchs breed and occupy mid to high latitudes of North America, which are shown in the areas, as I mentioned, in green and yellow on this map. But summer adults only live 2 to 6 weeks, and this is because they're breeding continuously, laying their eggs on their host plant milkweed. But as monarchs as a species cannot survive long, hard freezes, which is why, in part, we think they migrate as adults to avoid these low temperatures. So, migratory monarchs, in contrast, are those generally born as the last generation of summer or early fall, and as their larvae or caterpillars, declining temperatures and photoperiod trigger physiological changes. So they go into what we call reproductive diapause, which basically means that they're not sexually mature. They don't have a lot of eggs or sperm matured when they first emerge as adults, and they also have higher fat or lipid storage than summer monarchs. And both of these changes let them travel hundreds to thousands of miles to their overwintering destinations and live for 6 to 9 months, which is so much longer than their summer counterparts, which is kind of incredible for an animal that weighs less than a gram. So while eastern monarchs mostly overwinter in these high elevation forests of Mexico, western monarchs overwinter along the California coast. Both of these areas are the red areas on this map and also circled in blue. So here's the general timeline for Western monarchs in California overwintering groves. Migrant adults arrive to the coast in October and November, and they cluster together in groves of trees through the seasonally adverse conditions of winter, basically until it's warm enough to mate and lay eggs on milkweed. So it's generally believed that when they generally arrive around October, with peak overwintering season being around November and December, and the dissolution and beginning of breeding roughly begins in late January to early March, really depending on the how harsh the winter is that year. But basically, they're just waiting for those warmer temperatures to come back so that they can start producing those eggs and beginning the successive generations migration both inland and to higher latitudes following that milkweed. And a fun fact, it can take up to 3 to 4 generations for them to reach their highest extension up towards, the southern reaches of Canada. Just a quick note on eastern versus western overwintering habitats. So, though overwintering occurs both on the West Coast and in Mexico, there are some striking differences between the two locations' overwintering sites. So, for one, California's groves tend to be low, like below 800 feet in elevation. And that's also because, for the most part, and that's also because they're generally within a couple miles of the coast. Um, and this is really different than in Mexico, where they're relatively concentrated in these high Oyamel fir forests that are high elevation, around 9,000 feet. And also, the eastern population is just bigger in general, and there's only about a dozen sites or so that they are overwintering. So these are just have way higher concentrations of monarchs in these locations in comparison to California. In California, they are actually overwintering at hundreds of different overwintering sites each year, and they are actually spread out all the way from the north of the Bay Area, which is like the Sonoma County area, all the way down to Baja California, with some questionable sites even further north than that from our older records. But naturally, with this many sites spanning a wide range of latitudes, there's quite a bit of variation between them. They can vary in general size, the average population numbers these overwintering sites get, the main tree species they use for clustering that can vary a lot from where they are, thinking about North Coast versus, San Diego. Um, a lot of them can be found in more like canyon or ravine areas, while others I've seen, on hilltops. Some can just be thick windrows of golf courses, like this one you can see in Aptos, California. Um, and other ones are in contiguous native pine forests, like the one you can see labeled for Cambria. And other ones are just in state parks where people are regularly walking their dog, like this one near Steamer Lane in Santa Cruz, California, Lighthouse Field. So we're just seeing a lot of variation because they're using a wide range of habitats along the coast. And this is basically, just another view from the land of what it looks like in a pine forest versus another site that's not that far away that's a eucalyptus forest. And they both use these sites. They use both these kinds of sites. But unfortunately, the reason why we're so interested in monarch butterflies and what they use for habitat and where they're spending their time and how long they're spending their time is because their population is undergoing a pretty precipitous decline in the last couple of decades. So, as expected, there's variability in the estimated population size from year to year, which you can see on this graph. And these are the population counts that our volunteers collect during the Western monarch count period every year. But basically, you can see, though there is some variation, the number of sites monitored is that line in blue. So we've been increasing the amount of sites that we're monitoring, um, and that is still not accounting for the decline that we're seeing. In fact, a study done in 2017 by Schultz et al. Estimated, quote, an average abundance in the 2000s was less than 5% of the average abundance in the 1980s. So basically, that's a 95% decline since those earlier decades, which is, freaking us out a little bit. And this decline has come into even sharper focus in the last few years when the populations hit 2,000 for the entire state. So all those overwintering sites are only 2,000 butterflies in 2020. And then we had a couple of okay years. And then most recently, this year in 2024, we only had a little over 9,000 butterflies being counted in the entire state. So what that's telling us is that the decline is continuing, and we should be a little bit concerned at this point. And I think there's a lot of biologists that are really interested in trying to better conserve their habitat because of that. So, to better understand habitat use and protect migratory corridors and protect overwintering sites, we have to understand what they need to thrive. So that's just kind of like at the core of tracking animals is if we can follow them around, imagine if we could understand where all the monarchs are starting from where they start to migrate, coming all the way to the coast. If we knew where they were and how long it took them and what kind of habitats they're using, we could way better identify which corridors are the most important for them. Um, and same goes with the overwintering sites. We know that there's a lot of overwintering sites they use differentially throughout the season. And if we knew exactly what they needed from these sites, or how much nectar they needed, or how far they would travel for nectar, or if they move between these overwintering sites, we could better protect these locations and you know, provide better management guidance for people that are managing these locations like state parks and federal lands to make sure that they're in a state that the monarchs can thrive. But traditionally, to track monarchs, the main method used was sticker tagging. So you basically put a sticker tag, basically the size of my pinky nail, on their hind wing with a teeny tiny number that shows the unique ID of the monarch. And as you can imagine, these monarchs, they weigh less than a gram. They're super tiny. They're super fast, especially when they're warm. The recite rate, so the chances of you citing the same tag again is really, really low. So, especially if you're looking at migratory monarchs, for example, James and Capon in 2021, they reported that they tagged over 14,490 monarchs, and only 100 in 2022 of these tagged monarchs were ever recited again. So, it's an effective use, and we've proven that we can look at intersite movement between these overwintering sites, and we can show, a migratory monarch can show up at these overwintering locations, but we're losing a lot of information along the way. And I think for that reason, newer technology on how to track monarchs, like radio tagging, is a really, really promising new avenue that we've been looking into. Um, that I'm excited to talk to you about our work on today. So that was a lot of talking, so I just wanted to reward you with a pretty photo of some monarch butterflies that I took in Morro Bay, California, a couple years ago. I'm gonna take a sip of my coffee. Okay, so now we're moving on to how radiotelemetry works and just where it is relative to some other animal tagging techniques. So in an ideal world, we could just slap a GPS collar on all monarch butterflies and they could carry it and fly around and we could know their exact GPS location all the time. I would love that because we'd get really, really precise locations, and that's basically what we do with really big animals like deer, sometimes bears, wolves. Like GPS collars are really, really cool, and they're called GPS tags or satellite tags or collars because they're often around the neck of the animals that are bigger. And the reason why they're so popular and so amazing is because they're basically remotely sending off the animal's exact GPS location at regular time intervals. And that's the dream because you can basically get a really good picture of what they're doing, every day for kind of long periods of time. But the issue is, with doing this for monarchs, as you could probably imagine, is that GPS tags are really heavy. So not only do they have to have the GPS hardware and software, which is been getting smaller through the years, but they have a pretty sizable battery to power the device. So if you just imagine, like, if you want to say track a deer for a month and have to send a GPS location off 10 times a day, that's going to take a lot of power. It's kind of the same thing if you're using your Google Maps on a long road trip and you're using it all day, it's going to suck your power down really, really fast. And we can't just plug it back in when it's already on the animal. So that creates some problems, and it's just basically not feasible for little animals like teeny birds and bats and insects, especially while flying, because they have to carry that stuff around. So alternatively, for small animals, radiotelemetry is the most common kind of tag used, these radio tags. And this is because they don't require near as much power or hardware, so they can stay relatively small. And that's why they're really popular for tracking these smaller animals and what we want to do. With monarch butterflies. The only takeaway is that they're not giving you that exact precise latitude and longitude estimate that GPS collars are. But we're going to review really quickly how radio telemetry works in a nutshell with a silly cartoon I made. So, first, you're going to go ahead and put a tag on the animal. It has a little antenna to help send off that radio signal or burst of electromagnetic energy. It also has to have a power source because it's an active tag. That's the tag itself is what's sending off that signal. Um, this requires, some extra weight, especially if they're battery powered, but it can be a lot smaller. Um, and basically, what that burst of electromagnetic energy or radio waves is it encodes basically that unique ID. So it's just like the sticker tag. It's telling you this is Carl. We tagged Carl, and you're picking this guy up when we're scanning in this general area. Um, and different brands of tags basically just use different frequencies. So there's two really popular ones, and you don't have to remember this. It's 434 megahertz, and another one's 166 megahertz. And a new one we're talking about today is a different frequency. But these signals are basically sent out into all the area around the tag and in basically all directions. But biologists will then be listening with a receiver. And these are basically like a radio. You're tuning to the tag's frequency, and you're basically just listening for that corresponding frequency that that tag is pinging on. Um, and what's really cool is when you detect a tag on the receiver, it will display just basically two important pieces of information. It's going to give you that unique tag ID saying, hey, you picked up Carl and you're also picking up Susie in this area. And then it's also going to tell you the signal strength of each of the different tags that you're picking up. So it um, for this reason, you can actually sometimes use this to deduce how far away an animal is from the receiver, especially if you're waving like a directional antenna around, and you're getting different signal strengths depending on where you're pointing. Um, but signal can be affected by a lot of things, and this is basically just like, the radios we're using in our cars, and whatnot. Um, the distance the tag is from the receiver is going to affect that signal strength, so it's gonna decline the further you are from it. Also habitat, so if an animal is in, like, heavily forested habitat or there's a forest between you and the animal or a hill, that's gonna most likely block a lot of those signals. Also, the tag type, depending on the frequency, will have different characteristics. Some are better traveling through walls, while other ones are not as good at the short term wall traveling, but they can travel a much further distance. Um, and also just the antenna type and the direction you're pointing it. And last but not least, a lot of these tags, most of them are battery powered. And as that battery power declines, so does that signal strength. So that's another thing you have to think about when you're designing studies. If you have a tag, the battery life is really important for determining how long you can actually track these animals for. So there are two common ways the radiotelemetry is used to catalog the presence of animals. So they're basically two different kinds of receivers. And the one that I basically did a cartoon of is this handheld radio telemetry receiver. It kind of looks a little bit like a purse that you have a little sash around in a lot of ways. Like this is a low tech brand one, with that antenna. Um, and then the other one is called, you know, a stationary or station or remote tower. Um, and they have pros and cons to each of them, and in unison, they can be really powerful. So the handheld ones are basically like handheld radios, and they have that antenna, which you can basically use like waving around like a metal detector, but in the air, searching for picking up any tags in any direction. And doing this, especially if you're trying to get eyes and get an exact location of where the animal is, you can do this sweeping around to see if you're getting like getting warmer or colder based off the signal strength and what direction the animal is in. So that's really really nice, especially if you're trying to recite that animal and get an exact location. And depending on the study you're designing or the question you're asking, that can be really helpful. But the drawback is that you can't have a biologist out here 24/7. The batteries are going to die. They have to eat food. They are humans. So, if you're interested in when your tagged animals might be coming through an area, um, this is not going to be 24/7 monitoring. Whereas the remote tower might be really useful for picking up like signals 24/7 in your study area. So this remote or automated telemetry station is basically like a radio tower that's just always listening for those radio signals at the frequencies that those tags are pinging at. And just like radio towers, you try to put these in areas with really good visibility in all directions because those hills and trees are going to block signal. So you generally put them up in an area that um, the animal might pass through, and it's going to have good visibility to the areas that you think they are passing through. And then you're putting up, as you can see from this telemetry setup that Levi Souza of CDFW put up a couple years back temporarily. You have antennas going in multiple different directions. So that can be powerful as well because you can then write down which corresponding antenna has picked up a tag. So, say a monarch flies by, you can pick up that it was going from north to south or south to north. And you can kind of deduce what direction they might have been traveling in. And also, the nice thing is that a lot of these towers, especially the more permanent ones, have cellular capabilities. So you don't even have to go back out in the field. So handheld radio, you got to go out, you got to look for those animals. With these remote towers, you can set them up with a solar panel and a car battery, basically, with some other wiring and a computer, and it will remotely collect that data. And then, if it has cellular capabilities, basically send it off to you, and you can log onto your computer and see what it's picked up, which can be really, really powerful. The main takeaway is that they're just more expensive, more clunky, and like harder to set up and break down than just going out with handheld telemetry. So both of them have their own strengths and takeaways, but For both of these, if you with both of them, you basically are picking where you're surveying. So you have to know that or assume that the monarch is only going to stay in these areas. And that can be limiting for tracking migratory animals. So, say someone else set up a radio tower for a different study studying birds, and your monarch flew by and it got a detection on their tower. If you're just using basic telemetry towers, um, they will have no clue that that is your tag, and you might be losing that data. And likewise, you might be picking up tags that aren't yours, that are coming from a different study. And as soon as they leave that area, say it's like it's a monarch or coming from Canada going through, it just passes through once. You wouldn't get any data outside of the area that you're setting up these towers. And that's basically what's given rise to Modus. So now we're in the what is Modus phase of this talk. Um, and it was an idea founded, like a company founded in 2014 by Birds Canada. And it was basically researchers were thinking, hey, a lot of us, especially bird researchers that are interested in migration, we're all setting up these telemetry towers, but none of them are talking to each other. And if I get your data or you get my data, it just falls into the ether and it's hard to coordinate. So MODIS is the answer to that. And it's essentially a wildlife tracking system that is a network of interconnected radio towers set up all around the world. While there is, a lot in North America, there's actually a lot in South America, Central America, and even in Europe and Alaska. So it's getting really big. And what's really cool is you basically just go and register your tower. And as you get reports, it'll just report all the tags you detect, not just yours, to a central repository. And so it's ultimately just very, very collaborative. And the cool thing is that it'll pick up any radio transmitters from tagged animals that come within range, with a little asterisk being it has to have the proper antenna to detect that radio tag. Okay, so because birds migrate and Birds Canada set this up, this was originally designed for, looking at migratory animals, especially birds. It's become really, really popular, but also it's popular among other terrestrial animals. People are starting to tag a whole bunch of other things. I mean, we're tagging monarchs. Um, and there's interest in people tagging dragonflies, even bats. There's a strong bat community that's interested in tracking their migration using this technology. And because of this, the US Forest Service International Program basically gave us grant money to explore how to use this technology and how it could benefit monarchs. We're also working with really closely with Point Blue to do this. So now we're getting to our past work, so what we've done so far doing this, and the future directions that we're trying to take this research. Um, so it's really exciting, and we really want to go build these Motus towers to go detect monarchs, either migratory or moving on the coast. It could give us so much information of how they use their habitat, but we need to do our due diligence and answer some basic questions first. And so these three are basically we've outlined, like the first steps to intelligent tinkering is to like think through some of the basics first. So the first question we're trying to answer is, what type of radio tags are best for tracking monarchs? Some have different frequencies, some have different pros and cons. But whatever one is, we want to make sure it's the one that is best for monarchs and is going to be the most functional for them to fly with. And we want to figure this out first because that determines what kind of antenna we're putting on these Modus towers that we're interested in putting up. And second, we wanted to better figure out, as a partnership, Point Blue and Xerces Society, what the best protocol is for attaching these tags to monarchs. So it's the most non-lethal, and it's going to be, very. We want to make it as safe as possible for monarchs and have the least effect on their fitness. We're not trying to go out and sacrifice monarchs. We just want to study them. And last but not least, we wanted to look at this method using radio tagging at a smaller scale, looking at intersite movement at overwintering sites, to see if this is effective before we, shoot for the moon and scale up to try to retrofit a lot of Modus towers. Um, all over the place. Before we swing big, we want to see what it looks like on a smaller scale. And what you can see here on this map is basically just the location as of December in 2024 where Modus towers are. And so there's an okay amount of coverage, but there's definitely areas where we're starting to think about where Modus towers might be effective for specifically tracking monarchs, as well as other animals like bats and birds. And so I've been saying the royal we a lot. And it definitely I wanna highlight the fact that it's not me alone doing this, it's me giving the webinar, but we work super closely with Point Blue. They're our partners in all of this work, and we've been collaborating with California Department of Fish and Wildlife to get our permits and talk about best practices for tagging monarchs. The Nature Conservancy has been involved, Cal Poly, Environment for the Americas as interns that have helped us with this work. And, we have gotten a lot of technical assistance from Southern Sierra Research Station, which is another nonprofit, James Madison University people, and we got our funding from US Forest Service International Program. So it's truly one of the most collaborative projects I've ever worked on, and I'm really happy to have worked with a lot of the people that I unfortunately don't have cute photos of, so I just have their emblems. All right, so moving on to question 1. What is the best kind of radio tag that we wanna use on the mark? That was, like, first question that we really wanted to get into, and digging into the literature, there is some work that's been done radio tagging monarchs, before us. We aren't the first people that have thought of this, and, there's some work from, more of a Canadian lab, I think, and some other work from TDFW that I got to be a part of that was looking into, you know, what the best tag is and how to do this best, how to best attach these tags. And one that kept coming up was the low tech nanopin tag. And this is around 2022. And it was the lightest one on the market. It weighed 0.13 to 0.15 grams. Um, and it was battery powered. And if you reduce the number of pings to once basically every 30 seconds, 27.1 seconds, you could get a battery life of about a month, maybe more if you're really close to it. And one of the main pros of this is that this tag is actually on a frequency that's really commonly used on these Modus towers. So all those towers that are already up would probably detect these tags. And this is what we were planning on using at the beginning of this journey. But before I think we even got boots off the ground basically pretty closely after we started. This new tag technology came out that was really, really exciting to us. And it's because it's super tiny. And it's from a company called Cellular Tracking Technologies that we've been working pretty closely with. And it's called the Blue Morpho tag. I provided photos of here to show how tiny it is. And specifically, it's 0.06 grams. So it's so teeny small. I actually have one right here. I was gonna get out and show you and hold up. They're just so teeny tiny. It blows my mind and it scares the noodles out of me that I'm gonna like drop one and never find it again. Luckily, we have trackers, but it's still not super enjoyable to look for them on the ground. So they're just so tiny small. I don't know if you can see how tiny that is. Um, and the reason why they're this small and just 0.06 grams is because they eliminated the battery. It's just a little tiny solar panel that's directly connected to the hardware. So basically, when it's in sunlight, it starts sending off signals once a second when it's in full sunlight. Um, so this is how they're able to get it so tiny. So it pings only when the solar panel gets enough sunlight, though. So it's really cool. It makes it really tiny. That makes it a lot safer to put on teeny monarchs that weigh less than a gram. Um, and because it doesn't have a battery, it also would theoretically last the life of the monarch. There's no battery that's dying and then it's attached to the monarch, but it's not sending off any signals. So we're a little concerned that it's sunlight dependent. So, like, we want to make sure that it's still effective. And it also means that it has to go on the back of the monarch rather than the abdomen. But it's still really promising and really interesting. Secondly, there's a double-edged sword that this is on a brand new frequency. It's 2.4 gigahertz, so it's ultra high frequency, um, which is different than the other tags that are predominantly on the market. The biggest con of this is that it's not currently compatible. So that map of where all the these towers are up listening for birds and monarchs and all that stuff. This are these most of these are not yet compatible with this new tagging technology, so that would be something that we'd have to go in and what we're calling retrofit and add new antennas to these stations so they would be detectable. So that's a double-edged sword, but they are 2.4 gigahertz is actually the Bluetooth frequency, which every single one of us have on our phones. We all have Bluetooth antennas built into our phones, um, which means um, that the app that basically the Cape May Science Center developed in concert with cellular tracking technologies called Project Monarch app means that we can use our phones and citizen scientists could help look for these tagged monarchs potentially in the future when more of them are out on out on the landscape, especially the people that go out and count at overwintering sites. These tags could basically be detected. You don't need necessarily a fancy receiver. I do have a fancy receiver, and the people going out and walking transects looking for these tags as field hands would have those, but basically we could involve citizen scientists in these, which is really, really exciting for the future of how these movement studies could be taking place in the future. Um, that being said, they were so new that they had really only been tested by the manufacturer when they came out. So I think because they were so light, which is so beneficial to such a small animal, we are leaning towards using these CTT blue morpho tags. But we wanted to look into, um, the best protocol on how to put them on monarchs, and figure out how far we can detect them away from the receiver, and just kind of get the basic stats of how they work on the landscape. So next, our question was, what are the materials we need to tag monarchs, and what's the best protocol to use it? And so this is just a big shout out to California Department of Fish and Wildlife folks. You can see Hilary Sardinius and Levi Souza actually tagging a monarch butterfly with a nanopin tag right here. This is in December of 2022. And they did this to do a quick pilot study to look at whether they could track, site movement between small sites in a location in Montana de Oro State Park in California. And it unfortunately, there's some technical difficulties. Really big storms came through and took their towers down. So they only got a week of that kind of data. But it basically showed that it was feasible to track intersite movement with their pilot study. And they are some of the first people that um, I was in contact with that documented best materials and methods to tag monarch butterflies. And then the people that basically took that further, who have a lot of experience with butterflies, not just monarchs, for tagging and tracking butterfly movement in general, are Dr. Lee Brown and her graduate student Helen St. John, who's shown in the bottom photo. And they basically tagged a lot of monarchs with, the nanopin tags or 3D models and the CTT tags to compare how it might affect how they fly. Um, TBD on the results. I think there's a Monarch joint venture webinar that they will be sharing the results in this July, so stay tuned for that. But they've been sharing their results with us. And also on top of that, they've developed like really good best practice methods and materials for how to tag monarchs um, with these teeny tiny tags, and have come up with really good recommendations on what kind of glue you should use, et cetera, et cetera. Spoiler alert, it's duo active eyelash glue. So I bought eyelash glue to attach false eyelashes for the first time in my life to tag monarchs. So, you find yourself in funny situations sometimes as a biologist. But huge shout out to them. They really helped us. And but once again, because these tags are so new, in the winter of 2023, we wanted to go out and kind of refine that protocol since these tags had just come in the mail. And Lee and Helen were still refining their protocols on how to tag monarchs. Um, and we also wanted to answer some questions like, we're concerned this is only a solar powered tag, and monarchs cluster together with their wings closed in the shade a lot. So we were worried, is this even going to work with clustering monarchs? And we also wanted to figure out, if we're going to put towers to detect monarch butterflies, what is the range at which we need to put them near these overwintering sites to be able to reliably detect them. We don't know how far this detection distance is. So we just wanted to do some basic tests. And overall, it was quite successful. You can see us tagging, that's Helen, St. John, and Levi Souza right there, as well as myself helping tag a monarch butterfly. And then you can see the tag on a butterfly with its wings protected by glassine envelopes um, right there as the glue is curing. So overall, they're quite successful, the methods that we wanted to implement. And note that we had CDFW presence there to make sure that we were doing this legally. You have to have a scientific collecting permit to handle monarchs in the state of California. So we were doing this all above board. Um, but overall, these monarchs were safely tagged. And so we could now, write a more definitive protocol on how we would collect data on them. Um, we also determined that monarchs were detected successfully via not only the phone app, but the handheld receivers. And also, uh, we tested it by having someone hold a monarch with their wings closed in moderate shade and moving away from them with the receivers. And we did, it did demonstrate that we can detect them in moderate shade with their wings closed with these tags on, which was a huge win and gave us a really big green light on these tags and using them in future studies. We also figured, just with a really small sample size of four, um, selecting some of the heavier monarchs that we collected, that the tag and the glue weight was approximately, 13% of the monarch's body weight, which is pretty high, but a lot better than some of the alternative tags. So Helen and I basically walked around with sample tags at various heights, walking further and further and further away from each other with walkie-talkies and one of our handheld receivers. And I set one up so you can see what it looks like. So this is the receiver part right here. You can pair it with your phone, and this is the directional antenna. These are called elements. They just kind of concentrate the detection to just right in front of it. So you're basically using it like a magic little wand that you're pointing to sense the direction of the tags. Um, and so we were moving further and further away in a mixture of open habitat and forested habitat just to see, um, how far we could detect these tags from. And what we determined is that in forested habitat, which is where monarchs tend to hang out a lot, the detection range is it's pretty darn limited in comparison to open habitat. So you need to be within roughly 100 kilometers for those detections to get to the receiver and for you to record them. But in open habitat, we cleared, I think, a kilometer, up to a kilometer, we'd get a couple of pings. So now that we figured out the best protocol for collecting monarchs and tagging them, and we determined we want to use these CTT blue morpho tags, we also plan to do a study on Vandenberg Space Force Base. And so we wanted to do this smaller scale movement study, similar to the one that CDFW is doing, and track monarchs uh, habitat use and movement between overwintering sites. So it's really common. There's a couple places, several places along the coast of California where there are a lot of overwintering sites clustered pretty closely together. Um, and we just wanted to look in like how are these monarchs moving back and forth? How often are they leaving overwintering sites? Is there, a daily habitat range? We just wanted to look and see how they were moving around on this base. Um, and so our planned methods were to construct 2 to 3 of those temporary telemetry towers, and consider using other smaller remote receivers. And at those places, we would tag up to 15 to 30 monarchs, depending on the population size, at 2 to 3 of those overwintering sites. And then we would use these remote towers and also um, interns from the environment for the Americas, um, and they would go out as long as well as I would to do handheld telemetry at the nearby sites to go detect monarchs at all these different potential overwintering sites on the base. And what you can see here on the map, the blue is the outline of the base, and the red and orange um, points or polygons are overwintering sites. So they just have a lot of potential habitat that they could be moving between. So our aims for this future movement study was basically to better describe movement behavior of overwintering monarchs. Like I mentioned, look at the relative use of habitat outside these overwintering boundaries. How much time are they spending outside of them or moving between them, or the frequencies at which they're leaving them to maybe determine their home range. And also maybe identify really important movement corridors that are important to them, between these overwintering sites. We also are interested in looking at tracking the relative use of these spotty, seemingly low priority sites. So sites that, either only get big numbers of monarchs, every couple of years, or it's more sporadic, or sites that will see, activity earlier in the season, but then they disappear. We want to, in comparison to sites that regularly get high numbers nearby, we wanted to see if they were feeding into those, if they played an important role in the relationship to those climax sites, um, or if they really just weren't much of a priority at all, and weren't actually used that heavily. And understanding these dynamics would really help prioritize restoration and monitoring resources between the 30 plus overwintering sites that Vandenberg manages, because that's a lot of locations, that's a lot of effort. So figuring out which ones are most important for monarch butterflies, most important for restoration, could be really, really useful for their managers. And last but not least, if these CTT tags are very effective on monarch butterflies, this would really illustrate the value of retrofitting other Motus towers to detect these new CTT tags around the state of California. So we were really excited. This was set to happen basically last year, and we built the towers. You can see some Point Blue and Southern Sierra Research Station folks here. And we scoped out all the best locations. We purchased all the materials. We acquired all the permits needed to both handle the monarchs in the state of California, and also build the towers on Vandenberg Space Force Base. And we did, very detailed study design, trained all the interns, and this is what they look like. The topmost, that little cross forward, those are actually the antennas pointing in all the different directions that could pick up those CTT tags. And we couldn't really do the study. And this is because of the, as forementioned, really, really low numbers. There was no clustering that happened at Vandenberg. And the numbers overall in the state were so low that we just didn't feel comfortable tagging even a small percentage of the population this year. And we decided to push the study to the next year. So we're still planning on doing it on Vandenberg. We're planning on identifying some backup locations in case that happens again, or the population tends to swing north. There are other hotspots that we could similarly study. But we've been working on some other stuff too behind the scenes, aside from just setting up this small um, pilot study. And we've decided to move forward with working on retrofitting existing MODIS stations, which you can see on this map here in orange, to detect these new tags. It's pretty clear not only monarch butterfly people are excited about this technology, and it's been used not only by us, it's starting to be used by other people. Cape May Science Center has been doing tagging. I think they recently released a tag on the East Coast at Cape May Science Center, and they redetected it with a different kind of sensor in, I think, Florida. So they're starting to work. It's starting to be a proof of concept, and I think we need to start building out, these pre-existing MODIS towers to be able to detect these tags. Um, but also for other smaller animals like hummingbirds and bats, these could be a game changer for studying these small guys. Um, we're also prepping for the movement study, the pushing it for this fall, and we're also assisting in other tagging and retrofitting efforts. We have like a small group of people that we know are researchers working on this, and we all are sharing our experiences and resources in a working group. So it's been the most collaborative project I've ever taken place in, and there's just so much more to learn. And though it's really, really exciting to get super excited about all this tagging, there's just not a lot of tags out on the landscape yet, and all of these towers are not yet retrofit. So it's not quite time for us to be tagging huge amounts of monarchs, but I do think that the future uh, holds a lot of promise for using this technology. First, I think it's going to be most important at that smaller scale. I think it's going to be really exciting for Western Monarch Count volunteers. If we do this study in an area, we will let them know, hey, download the Project Monarch app. Um, you could potentially be detecting these individual monarchs on your phone, and you can even name them, which is really fun. So you could detect a Carl or a Sharon yourself. Um, but, as far as this goes, science is slow, but we're chugging along. And, the best way you can help yourself is either if you're on the coast of California, which I'm sure not all of you are, you can volunteer to help count these overwintering sites. Scientists and researchers depend on these to figure out how well the population is doing from year to year. Or you can join the movement and donate to Xerces to help fuel this work in the future. And I just really want to thank all of the donors that have so far. We really rely on your support. And with that, I will take questions.
Thank you so much for that, Ashley. Yeah, this research is just so interesting. And yes, we do have a lot of questions coming in.
Okay.
So we
I felt like I just talked a lot and I like should have paused for more questions for people to stop and catch them
no,
all.
no, no, no. Don't worry, don't worry. Yeah, so we'll try to get to them all, but I don't know if we'll be able to. But we'll just dive right in. So going back to the beginning of your presentation,
Yes.
some of the background about monarch populations.
Yes.
We had some questions about what happened, if you know, in 2009 and in 2018, 2019 for those incredible declines in the monarch populations.
Yeah, unfortunately, no matter what, my answer is gonna be a little hand wavy, like, no one really knows. We have a really good blog article on the bounciness of butterflies, so there is a natural fluctuation that happens with these, like, our selected species, these species that lay thousands of eggs and have, like, really we can have really good years, and they could have exponential growth, and we'll have really good years, and then other years where basically either the emergence of milkweed isn't timed well with when they're ready to start reproducing, or there's a massive heat wave or something that can knock out a big portion of the population, and especially if they're in a vulnerable state, like the last generation, the migratory generation. If the big portion of them gets knocked out, you're going to see lower really dramatic declines some years. I think what's really scary is that when there were millions and there'd be a big decline, it's scary but not terrifying, like going down to 2000, like you mentioned in 2020. Now, we don't have direct causation, but we do have some hypotheses. Like, I think one of the hypotheses, and by hypotheses, I mean like there is not direct evidence, but like there's really strong heat waves, and monarch butterflies have an upper thermal limit. So do their larvae, and sustaining like really, really hot heat waves that, like, I remember were hitting our inland areas really, really hard in the late summer, right when we think that they might migrate. I think just timing of bad weather can really affect and wipe out just a big portion. Not saying that they can't bounce back, but that does lead to these big bounces. And just considering that there's a background amount of decline because of a lot of the risk factors like pesticides and habitat loss and all this other stuff, it's just becoming more and more dire when that happens.
Yeah, that makes a lot of sense. Thank you for explaining some of those fluctuations. And we will try to get that blog post link posted in the chat for everyone if they want to take a look at that and read a little more themselves.
Totally. It's written by one of our collaborators. She's really smart. She's the one, I think she might have taken hand in that analysis I mentioned. So it's a really good read.
Perfect. On the monarch decline, while we're still on that subject, if you can speak to, is there a difference in the monarch declines between Mexico and California, if at all? And similar question, what might be the cause?
Totally. I'm less up to date on the effects of the eastern population. I know for a while deforestation was a really big like issue, and they were worried that they were going to lose the locations that they were overwintering. And then I think later in the 2000s, um, it was really identified that GMO and Roundup Ready crops were a huge played a huge factor in the eastern monarch declines. And by that I mean like there's a lot of homogeneous cornfields that go in, but what a lot of the cornfields, and I like hope I'm like repeating this right because I read this like a decade ago. Um, basically, they are resistant to Roundup, and then they would basically spray Roundup and get rid of all the milkweed that tends to grow on these edges. And so basically, it was just a mass removal of a lot of milkweed on the landscape. Um, so they think that that mass removal of milkweed might have played a really serious role on the east. It's not as clear what are causing the declines in the west, but in general, we're really concerned about broad use of pesticides. It's a bit of a pesticide gauntlet if you're thinking about the Central Valley of California, because that's where all our agriculture, like all our food we eat, we need that food. But I think the prevalence of pesticides there, and also year after year, we watch, overwintering sites get destroyed. And so there's just less and less habitat for them to come to overwinter.
Yeah, that makes a lot of sense. Speaking of, since there's a decline in the populations, Holly wants to know, are there efforts to increase the population via human efforts?
Yes. Oh my gosh. Yes. Well, we have habitat kits, which is really cool, Xerces. So if you have land that you want to, and I don't know the details of it, but basically, if you want to plant nectar habitat or pollinator habitat, monarch butterflies like flowers, but so do bees and our other really important pollinators. You can go and put in, pollinator friendly habitat landscapes. Also, there is WAFWA, so basically a conglomeration. A lot of the Western states' departments of fish and wildlife came out with this plan that they're now revising for a new update that we're helping with, that identifies all the research needs and the conservation needs that they want to target to enhance uh, or monarch butterfly habitat in the West. And different people might be reaching for different goals, but like one of our goals, especially as an overwintering monarch specialist, is increase the signage, increase awareness of a lot of these overwintering sites, and prevent more of them from being lost, because a lot of them just get developed or cut down and people just don't even know that they're overwintering habitat because monarchs are only there for a couple months of the years, and so it's just really easy to miss it. And so I think just increasing that visibility is really important. And we have a lot of resources on our westernmonarchcount.org, on some other efforts that are going on.
Yeah, we'll be sure to post that link in the chat as well. All right, getting on to some of the tagging and research. Someone was curious if this is the same methods that they use in Cape May, New Jersey. And I suspect that might be about the Project Monarch. And so go
Yes,
ahead, tell us more.
totally, it is. So we were in talks with those are basically the company that makes cellular tracking technologies. They work hand in hand with Cape May, and they're the people that basically Cape May developed the app to detect them, like citizen scientists detecting them, and uh, CTT are the people that developed the tags. These are the same exact tags, and this is, I think, basically there's like a very small group of people uh, that are very, very excited about these tags that are like basically taking it and running with it. But those are the same exact tags, and we get together occasionally. Some people from CTT or Cape May will join our group, and we kind of exchange like, what have you been up to? Like, how did it go? So we are in contact with them. Yeah.
Exciting. Yeah. All right, speaking of tags, this is great. Each question is just like segueing
Perfect.
into the next so perfectly. A lot of participants want to know, how much do each of these CTT Blue Mofro tags cost?
That's a great question. I think it had to go up. The price did go up recently. I think it's about $200. Um, I can fact check that, but I think it's around $200 now. So they are not super cheap, but they're not terribly unaffordable if you're comparing the cost to other tag types. But
Good
yeah,
to know.
not cheap.
Yeah. Similarly, can anybody purchase them?
Yes, so anybody can purchase them. I think the emphasis, and I have like uh so many garbage slides that I can poke through. Um, but this is a slide on legal protections. So anyone can purchase these tags. But in the state of California, you have to have a scientific collecting, or like a specific use SCP scientific collecting permit, to handle monarchs at all. So in the state of California, you can't just tag monarchs. You have to have a permit. And it's kind of cool because it protects, there's a lot of clustering monarchs here, and we don't want anyone like, you take a net and you wait for them to get really cold when they're clustering together. So we basically cheat. We wait till they're paralyzed and like too cold and tired. And then we go and we gently shake a couple like of a small cluster off. And that's how we collect them and keep them in a cooler before we process them and tag them and take all their metrics. Um, but we don't want, like, anyone doing that, because if, say, 100 people decide that they're gonna go do that, like, that might be detrimental. Um, that being said, there are no federal protections. This is kind of an old slide, but they're basically proposed to be listed in 2024. That those protections don't go into effect until it's completely accepted, which they think it will take about 12 months, but I don't know how long that's actually gonna take. Um, and they're seeking comments right now. So, theoretically, if it's any random wild caught monarch outside of the state of California, you could theoretically tag it and then see where it goes and see if it gets picked up. With the new federal protections, the one thing that they do prohibit is disturbing clustering monarchs, I think. But captive rearing and I think capturing, I don't quote me on this, but I think it's like captive rearing, you can do up to like 250 monarchs. So you can do small stuff with these tags outside of the state of California. That was a very long answer, but I hope that helped.
No, that's okay. That's good to know and important information to make sure that they're protected while we're studying them.
Totally.
Similarly, do you know the approximate cost to retrofit the Modus towers? A lot of people are interested in that.
Oh my gosh. Oh, that's such a good question. I should have like looked up the costs. It's not super expensive materials wise. I want to say it's, and I can follow up with this and you can like, follow up with everyone. I want to say it's in materials wise, definitely less than $1,000. Like probably closer to $500. I want to imagine maybe even less, I think. The main issue is that a lot of these locations, they're picked to be remote on these high areas, so getting to them and doing the actual retrofitting, you have to, like, pull down a tower that's, like, 30 to 40 feet high, so, like, getting all the materials, getting the power tools, and then, like, retrofitting it, I think is the biggest cost is, like, getting people out there, so that's why we're focusing on retrofitting already pre-existing towers and working with people that already have the skills and the know-how to do it. And so if they're just going to go do tower maintenance, because you have to go out and make sure it hasn't blown over in like a big windstorm, they have to check on these things occasionally. We go, hey, here's all this material, like we already paid for it. Do you want to put this up? And so they retrofit their tower just when they go out there, and we get more towers that can detect these tags.
Yeah, bit of a trek to get to some of them, I imagine.
Yes, pretty in a lot of places, but hard to get to.
Yes. Okay, continuing on the tags.
Yes.
Denise wants to know, did the blue morpho tags interfere with the mating process?
Oh my gosh, Denise, that is such a good question, and I didn't want to totally blow up, Lee and Helen. We were worried about that. We thought about it, and the ones that we originally with low tech tags, like the first iteration that we did with CDFW and that they led, um, you know, other studies kind of put them on the abdomen, and it did look like it might interfere, and that was like not great. But now that we are putting them on their dorsal thorax, um, we think that their abdomen could be a little bit of like could curve and actually do the mating. But it wasn't confirmed. But you should watch the MJV July webinar and look at what Lee and Helen have done from James Madison University. But they actually tested that and they can mate. So major props to them and keep your eyes open for news on their work.
Excellent, excellent. Good to know. All right, a couple similar questions on this subject. Have you recovered any tags, i.e., after the monarch expires, or found tags that have become unattached? And then related, someone was also wondering about removing the tags, or is that not something that's done?
Totally. So we're hoping that, and like, so I wish I had more answers for you. Unfortunately, we did that, like, 4 monarch test, and then basically there are no monarchs, and we didn't feel comfortable, like, tagging the monarchs we did find. So we don't have a ton I don't have a ton of data on this. That'd be a great question for Helen and Lee, who did more work. on this so far, but um, for the low tech nanopin tags, I swear to swear to all that is good, there was one that fell off during that study because I would go back out and handheld track, and it was somewhere on the ground, and I could not find it for the life of me, but it was on the ground. So there's no way that that was still on the Monarch or the Monarch had died. And that was those battery powered ones that were on the abdomen. For the CTT tags, um, theoretically, it would last the life of the Monarch. So the Monarch would probably die with it still on it and probably away from the site if they are beginning that remigration inland. So it'd be away from at least our focal sites. Um, Ideally, we would recapture it. It would be really cool if we could get it all the way from when we tagged it to when it had died. That would be a total goal of ours. I have noticed, unfortunately, these are solar powered tags and they tend to be bottom heavy. So when I have dropped them before and they always flip upside down and it kills me because that turns the pinging off and then you're in trouble because they literally are like this smaller than my pinky nail and dark. So yeah, you could theoretically find them again, but you just really, pray that they do not flip over.
Yeah, that would seem, talk about needle in a haystack, but tiny, tiny tag in a field.
I had an intern and we dropped one while we were doing like range testing of these towers that we set up. We wanted to collect and see how well these tags were being detected as we moved around them. We lost one and it took him like 3 hours to so yeah, it's you don't want to drop them.
Yeah, definitely.
Yeah.
Well, on that, and a little bit more about the Monarch and the weight of the tag.
Yeah.
Someone wants to know, they said that they think you mentioned that weight of the tag is 0.06 grams, and the glue comes out to about 13% of the weight of the Monarch. Assuming that the glue is really light, they calculated that the average weight of the Monarch is then a little over zero point 0.5 grams. Is it common for your monarchs to be above zero point 0.5 grams? Is that a
Oh,
typical size?
I think the US Forest Service, I like look this up to make sure and we only tagged like four, but in my experience, it aligns with my experience. Um, They are about a half a gram. So they generally weigh about a half a gram, but they can range from like they can be a little smaller to a little larger. We used a cutoff from Wilcox et al. 2021, a different paper basically that said we're going to try not to tag any monarchs that are smaller than I think it's like 0.35 grams ideally. So we do have a lower cutoff. I need to triple check what we wrote down. I think it's point 3.5 grams at the very lowest. And we're trying to get these heavier monarchs that might bias our data, but we're just trying not to, unduly burden smaller monarchs with these tags. And the 12% of their body weight that we calculated, that was the tag and the glue on top of the monarch. That ended up being about 12% of their body weight of the monarchs that we specifically tagged.
Yeah. And you said, I think I remember you said you tried to find the largest ones that
Yes,
day
we
for
tried to get
testing. Yeah.
yeah, so we, like, do the collection and you end up getting, more than you plan on, and what we do I should have explained this better. We put them in these glassine envelopes, which basically protect them in their wings, and then we put them in a scale that goes to the thousandth gram because 'cause they're so tiny you can't use normal scales with them, with a little wind protection because 'cause they're so sensitive, and we get their weight before and we'll do that with more monarchs than we plan on tagging, and then we immediately release the smaller ones. If they, like, don't hit our criteria or we have plenty of larger ones, we'll just go with the biggest ones, and then we'll take those after we record their weight and then attach the tags to those ones.
Yeah. Thanks for explaining that a little more.
Yeah.
All right, switching gears here. Someone is asking, they see notifications where parks want to remove non-native eucalyptus trees. Is there any check or concern whether they could be overwintering sites for monarchs before these are removed?
Yeah, so I think one of our biggest objectives, like I mentioned, is to, at least for known overwintering sites, increase the visibility and, make sure that the important land managers, like, um county commissioners, like, city people that approve building permits, like, we've done that meetings with UC like, Santa Barbara folks, and state parks people that they know if they have habitat. Generally speaking, before major habitat changes happen, California has this thing called the CNDDB, and it has theoretically all the known sensitive sites. And so they should theoretically be checking that and know that it's there. Accidents do happen, but they should be checking that. And also, if you're concerned that someone might have, missed that that's critical habitat, you can check out our overwintering sitemap on our website and you can compare. So if you see that like they're getting ready to do a big development in where eucalyptus groves are, you can check the map and see if that's an active or known overwintering site. And if you see clustering monarchs and you see that it's at a not defined overwintering habitat, please email us. We'd be happy to go out and check it out, and add a new site to our list and try to protect it.
Great. Thanks for that. Yeah, we'll make sure that we get all of these links to everybody.
Yeah, sorry, I'm dropping a lot of.
No, 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 that's okay. Okay, couple time for a couple more questions. This one's really interesting. Tara wants to know, do you prefer females over male monarchs for the tagging?
That's a good question. In general, I think we were kind of shooting to go 50-50 male to female just because there might be differences in how they move. And that would be interesting if we got a high enough sample size to look at both of them. So generally speaking, I try not to be too biased to one gender or another. But yeah, good question.
Yeah, similarly, have there been any studies that compare the strength of flight in monarchs who have the low tech tag on the abdomen and the CTT tag on the top of the monarch, similar to comparing males versus females?
Yes, you guys are asking all the right questions. This is exactly what we were interested in, and that's what Helen, and Lee from James Madison University are working on. And they did some pilot work, basically looking. It was summer monarchs, but they tagged them either with fake tags that were like 3D printed versions of those because these tags are not cheap. But they compared the two, and I think they're still analyzing the results. So I don't want to like blow up their spot. But I think if you are interested in the answer, come to that webinar in July that MJV Monarch Joint Venture is doing because they're going to talk a lot more about it. I'm going to spend like 10 minutes doing similar spiel to what I did here, but I think the real star is their work that they're doing.
Thanks. Yeah, really interested to attend that one as well. Okay, time for like two more questions here. There's so many. Unfortunately, we will not get to all of them. But Deb comments that they had a lot of monarchs staying in the Bay Area this winter in their gardens specifically, but also in a number of other gardens. So they noticed a lot of late caterpillars. Do you have any thoughts on whether this has been seen elsewhere this winter?
Oh, okay. I don't know about this winter in particular. Sometimes, we have one of my colleagues checks iNaturalist to like see the rate of caterpillars that are being reported at different times to get our finger on the pulse of that. I haven't checked in with her recently, but basically Southern California, that's been reported since either the late 80s or 90s that there is a small proportion of monarch butterflies on the coast that do reproduce through the winter. So not all of them are in like full reproductive diapause. You know, it's nice to paint a pretty picture to say that all of them, like, perfectly with reproductive diapause, show up, don't do anything, and then leave, but some of them come out of diapause, earlier than we think. Some of them don't even go into it at all, especially if they're not coming from those higher latitudes. So, there's a lot of activity there, and there's also been really well documented activity, especially, I think, in the 2020 year of a lot of reproductive activity, specifically in the Bay Area, and one of the things that, definitely, like, encourages that, which we're not so sure is good or not, is non-native tropical milkweed. It doesn't die back. So a lot of the native milkweeds in California, excuse me, I have to cough. Excuse me. Sorry. Um, a lot of the native milkweeds here in California, they naturally die back in the winter. And we think that that is when monarchs also come together and then they do their overwintering thing and they cluster together. And then as it reemerges, that's another signal for them to break out of reproductive diapause and begin the breeding and the remigration. So tropical milkweed, there's one study um, that shows that there's evidence tropical milkweed exposure might kick them out of reproductive diapause or might encourage breeding even in the overwintering season. So in general, like the jury's kind of out, but Xerxes' stance is basically like, hey, let's stick with native plants. Let's stick with what we know, what has historically been here. And rather than plant tropical milkweed, plant native milkweed, let it die back. And that also prevents diseases like OE from like getting to really high infection rates as they build up on those plants rather than letting them die back and zero out those disease rates that can build up on those plants.
Yeah, that would be really interesting if there is a connection there. Um, and again, sound a little like a broken record. We have a lot of native plant lists and specifically monarch plant
Yes.
lists on our website. So Carly or I will get all those links either to you all in the chat or when we post this webinar later to YouTube, we'll make sure to include a lot of the links for some of this research and all of that. So if we can't get to all your questions, don't worry, we will get you all those resources. But on that, last question
Okay.
is how can folks get involved in these monarch counts or other monarch conservation or science?
Oh my gosh. Also great. There's so I'm like, I'm thinking of all the links we could send you also. Um, depending on where you live, I would recommend different things. If you want to dedicate some significant time and you live near the coast, please join the Western Monarch Count. We need volunteers. We need, there are literally hundreds of sites. There are 4 people on our team, but we have a legion of like amazing volunteers and coordinators that go out. They look for these monarchs. They report them through Survey 123 app, and you can learn how to report them. And we rely on those numbers to understand how well the population is doing. So the first thing you could do is either join the Western Monarch Count if you're on the coast. Secondly, there are a couple of projects where we don't understand, which is why we're trying to tag them. Um, where they go once they leave overwintering sites, like that first generation, where are they going? What kind of milkweed do they need? And so there's the Monarch Mystery Challenge, that basically you can report either via iNaturalist or a different link. If you see monarchs, if you see them, breeding on milkweed, you can take a photo and report that. Like that's a pretty low effort way that you can help give us data. Um, and that's a really, really useful thing. If you want to also just with the plant lists and all that stuff, creating a pesticide free pollinator friendly space, those gardens, that's a huge deal. They need more habitat. When we develop housing or when we like clear things for agriculture, um, it's taking some, resources away from them. So it's really nice to be able to give that back. And also you get really pretty flowers. So highly recommend, planting a native pollinator garden. And if you don't have time for that, you can, donate and help propagate one of our programs and have someone else basically do it for you, which sometimes I prefer, when you're too busy.
Thank you for mentioning all of those. There's a lot of good options for people who have different time commitments, different
Yeah.
resources available to them, but we love everyone's passion. All right. Well, we are out of time, but I wanted to thank you, Ashley, so much for joining us. This was so fascinating. And thank you for all of your hard work. Also, wanted to mention that if folks are interested in learning more, Ashley was featured on a recent Bug Banter episode, the Xerces Bug Banter podcast. So you can find that on xerces.org/bug banter. And episodes come out twice a month. So whether you listen to this one or another one, please subscribe wherever you hear your podcasts and hear the latest episode. And then they're also available on YouTube, as this webinar will be as well. I also wanted to thank our audience for joining us today. We do hope to see you on our upcoming webinars. And finally, thank you so much, Carly, for your help today. All right, everyone, have a wonderful rest of your day. Thanks again for joining us.